Thank you very much for the introduction. I'm sharing my screen. Is this all good? So I would like to thank you, the organizers, for the very kind invitation. It's a pleasure to be here and to give this presentation to discuss a bit of our recent results. So my lab works on gene regulation, and we try to infer gene regulatory networks from single cell data, where we are aiming to find key regulators, transcription factors for each cell type in heterogeneous tissues, but also in dynamic processes. And what we are mostly interested in is to find the enhancers, the regulatory regions in the genome, where these transcription factors, usually in combinations, are binding, and to understand a bit better how these enhancers work and what their architecture is. So we're applying this single cell regulatory genomics, as you could call it, to a variety of model systems. About half of the lab works on Drosophila, where we are studying eye development and also the brain, as well as other tissues in the context of the fly cell atlas. And on the other hand, we work on mammalian enhancers, where we use usually in vitro system cell culture, and since recently also organoids. But this talk, I will focus on Drosophila for you. So since a few years, it is possible to profile chromatin accessibility at the single cell level, particularly with single cell ATAC-seq. So using the TN5 integration that happens mostly in accessible chromatin, this method is also applicable to single cell data. And even though at the single cell level, this technique provides relatively sparse data with only a read that covers an accessible region in a single cell once in a while, even though that is sparse, if we can group single cells based on this sparse data into similar if we can put the similar cells together in clusters in cell types, then by aggregating this signal over all the cells of the same cell type, we can find regions in the genome that are specifically accessible, such as here in the green cell, specifically accessible, and that may then point to regulatory regions such as enhancers, promoters that are used specifically in that cell type. So we applied this technique, single cell ATAC-seq, to the eye development in the fly, as well as to the brain, together with single cell RNA-seq. And in total, this is more than 400,000 cells already. And I will discuss both of these systems here. So the first one on the iDisk, this is published data since a few months. This came out in molecular systems biology, work by Carmen and colleagues. And then the brain data that I will discuss is unpublished. The eye is interesting for me to still, even though it's published, to still explain, because it allows me to also go over different methods that we developed in the lab, such as scenic and cystopic. So let's start with the iDisk. So this is an epithelial tissue, two-dimensional sheet of epithelial cells that is floating in the larvae, and that will develop into the antenna, the eye, and a part of the head of the adult. So here you can see on the left a t-SNE of the single cell RNA-seq data that we obtained from this tissue, and on the right a t-SNE from single cell ATAC-seq data on the same tissue. And so what was nice is that we could already find different clusters that represent the different known domains and cell types in this system. So the advantage of this system is that all the cell types are already known beforehand since many years. And so we use it to test new technologies and to test new computational methods. So for example, here in the RNA-seq, we find 3 clusters for the antennal disk, A1, A2, A3, and Arista, which corresponds to the different rings here. And in the eye part, we find a trajectory from progenitors and precursors in here anterior to this furrow over the morphogenetic furrow in green, and then more and more towards the right here, posterior side, which are differentiating photoreceptor neurons. And the same is found in the ATAC-seq side, although here one of the antennal rings is split into 2, and the rest is agreeing very well. So how do we know which clusters represent which domain? As you can imagine, this is based, like usually, on known marker genes. And here's just one example of Distalis, which is expressed in the 2nd and the 3rd and the arista, but not in the first antennal segment. And that's, as you can see here. And so that's why we are able to find all these clusters back. So now, because these clusters represent this spatial organization so well, we could actually draw a virtual template, a virtual tissue, and then place the centroids of all these clusters into that tissue, and then project all the cells in there while preserving the distances between these cells. And this allows us now to look at gene expression a bit more realistically. This is atonal and senseless. You can see them in the t-SNE and then also in the tissue, and this agrees with how it's supposed to be expressed. Atonal here in red and senseless in green. And here's some other examples like gut, optics glass, homothorax, and dachshund, which agree with antibody stainings too. Then we could do the same with the attack data. So the single cell attack data is also projected into this virtual template so that we can look at chromatin accessibility in this tissue. So here's an example of a region in yellow here. This is a region upstream of the gene delta. And as you can see here in all these tracks which are generated by aggregating the cells over the different clusters, this region is only accessible in the second antennal segment. And so that's what we can see here. Now the beauty of Drosophila is that we can actually test these regions for enhancer activity using transgenic flies. So when this region here is cloned upstream of GFP and integrated into the fly genome, we can actually see the GFP expression to be specific indeed to the second antennal segment. And this is another example for a region near 7-up, which is accessible only in the photoreceptor neurons, and indeed the GFP is produced only in the photoreceptor neurons. And this is true for many other regions that we tested, for which we used the lines from the Flylight database at Genelia Farm, where you can see regions that are accessible in some domain, and then the GFP production is in the same domain. And this progresses more and more posterior, as well as in the antennal rings. The attack set can be used to predict enhancer activity quite well. So now, further, we can ask, can we find for each of these cell types, now we have the RNA data, we have the attack data in each cell type, can we find relevant transcription factors? So if we use the single cell RNA-seq data, we can use a method called scenic that was developed in the lab by Sarah. So I'll go through that briefly. So in scenic, we first define modules of genes with an upstream regulator transcription factor. So these modules are built using GeneTree or GRM Boost. This is a random forest regression method whereby the genes are predicted target genes of the TF because there is co-expression or co-variability, this can also be nonlinear, between the transcription factor and the target genes. And then in the second step of scenic, we use all these gene modules and do motif discovery in regions. In the fly, we use 10 kilobases around the transcription start site. In human, we use 20. So it's still relatively close to the TSS, but in quite some domain around the TSS of all these genes, we look for motifs. And if we find the motif of that regulator to be enriched in this gene set, then we keep this and we prune it further to keep only the direct target genes with the motif. And then we arrive at what we call a regulon. And then we score again all the single cells with these gene sets, which are regulons of a TF and their predicted target genes. This method now also works with ChIP-seq tracks from ENCODE, for example, where not only we search from enriched motifs, but also enriched ChIP-seq peaks for transcription factors. So this is the result on the iDisk. So here you can see the transcription factors that are found with regulons, and these are all the cell types. And so, for example, in blue here, we see the atonal regulon to be active in this morphogenetic furrow. This has 106 predicted target genes. Here's the expression of atonal in the second antennal segment and in the morphogenetic furrow. And this is the activity of that regulon, of this whole gene set, entire gene set in that tissue. That's with RNA-seq. What about the single cell attack seq? That we can also use to predict transcription factors. But first we have to go through a modeling approach because, as I explained to you in the beginning, this data is very sparse. And to analyze that, Carmen in the lab developed a method called systopic that uses topic modeling. This is borrowed from the text mining field. Basically, it's a latent Dirichlet allocation or LDA approach, that is going to find topics. These are latent variables, and this is a probabilistic framework. And these topics are now representing probability distributions, 2 of them, that after optimization will have the probability of each region, how much each region likes a topic, and also a probability distribution over the cells, how much each cell likes a topic. So after this optimization, this yields 2 matrices. One has all the single cells and how much they like each topic, and the other matrix has all the regions in the genome, all the peaks, and how much they like the topic, like each topic. So the first matrix we can use to cluster the cells, and the second to cluster the regions. So here's some results on the ion tunnel disk for different topics here. Every column here is a topic, and you can see the cells that like this topic. So these are the cells that are anterior to the morphogenetic furrow, the progenitors. And this now here is a t-SNE of all the regions in the genome that have some signal. They're clustered by the topic, by their topic loading. And in this cluster here that is red, these are the regions that belong to this particular topic. So these are the regions that are accessible in these cells. And then we can see other topics where cells are a bit more progressed to more and more differentiating photoreceptor neurons or in cone cells. So what we can do now with these regions that are co-accessible in these cells is to do motif discovery on these small regions. And indeed, we find then relevant transcription factors. In this case, optic oculus is a factor that is known to operate in these cells, and grainy head. And then later in the morphogenetic furrow, we see atonal appear, and glass more later on. We can also validate this experimentally. So in this case, we focus on this topic here, anterior to the furrow, and we actually used a fly line, a genetic driver that expresses GFP only in the domain anterior to this morphogenetic furrow, so only in the domain of the progenitors in the eye disc. And then by fact sorting these GFP positive cells and performing bulk attack-seq on them, we could find the regions in the genome that are specifically accessible in this green domain and map them to this enhancer t-SNE. And indeed, they are the regions that we had already found by single cell attack-seq. So this could be validated in this way. All right, so this is now 2 approaches, RNA and attack, so it becomes a bit confusing maybe. And so we need to find ways to exploit both layers. And in the brain, I will also come to that. So the first thing we tried in this project in the iDisk was to make use of, well, basically to solve an important issue when we want to link them, and that is, how do we link regions to genes? And for that, we are using this common latent space where the RNA and attack are projected on. So now we have, both of them have the same dimensions of virtual cells. This would work even better if you would have multi-ohm data, single cell multi-ohm. This is a new, relatively new 10x kit that produces both of the same cell. And then we use a technique, again, Genie3, in fact, that was also used in the first step of scenic. But here we use it to predict which regions could regulate which target genes, basically using the covariability of the attack signal in a region with the gene expression of a nearby gene. And this allows us to go very far around each gene and fly to 100 kb, so that would be 2 megabase in human, and link regions to target genes by this covariability. So if a region, a genomic region, is accessible only in this domain in the antennal disk, and the nearby gene is only expressed in this same domain, then the weight of this link will be very high. And this way, this is all linked. So here's some examples of a gene, scabrous and fill, and the regions are linked to it. And so now we can prune the scenic regulons, in this case, the atonal regulon, by using only edges in this network that go from the factor to a region that is co-accessible where this factor operates. And that region is linked to the target gene. And so this is now the pruned higher quality regulon. Now we have these gene regulatory networks from attack, from RNA, and we could go one step further and investigate the architecture of the enhancers to see if we can find combinations of motifs. there. So for that, we use deep learning. So Ibrahim trained a convolutional neural network on sequences and predict in which topic, in which domain they are accessible. And this works quite well. And so what we can do with this model is to ask then, after it has been trained, what are the convolutional filters that are important for a certain region to be predicted to be accessible in a certain domain. And in this case, for the atonal topic, we find the atonal binding site, which is a BHLH factor, as the first most important filter. And we can backtrack into this model to predict which nucleotides of the enhancer were informative to make that prediction. So I see that time is flying, so I will skip actually the next 2 slides and go a bit faster now to the brain. So this is an overview of the different analysis we did on the iDisk. Now let's go to the brain. So this is another cup of tea. Here we are dealing with much more unknowns and a much higher complexity. We had published in 2018 this atlas of single-cell RNA-seq on the fly brain. And since then, we have expanded this to 3 times larger data set, where now we can find many more cell types in the brain. And these clusters here agree very well with specific GAL4 lines that have been generated at Janelia Farm that label specific neuronal classes. So this is an example of an optic lobe neuron, the T4, T5 neurons that have been FAC sorted at Janelia Farm with bulk RNA-seq. And this signature actually maps very well to the single-cell RNA-seq clusters. So single-cell RNA-seq in the fly brain is also able to find clusters of true cell types. And we could label already or annotate already many of these clusters, especially in the outside of this. In the central brain, in the middle here, is much more difficult to annotate. Okay, so now to single-cell ATAC-seq on the same tissue. So since we have published the RNA-seq, we have been working the last 2 years or a bit more on an atlas of single-cell ATAC-seq in the fly brain. And we decided to profile the brain at different developmental time points, from the larval brain, where neurogenesis is happening, all the way over pupil development, synaptogenesis, and the adult, where we have the most cells, here where we have all the mature cell types. So for this talk, I'm going to focus on the adult. The development will have to be another story, another time. So in the adult, we have found, after a cystopic analysis, quite specific clusters. And this is the RNA-seq data. And we were able to link many of these clusters to specific clusters in the single-cell RNA-seq data, and thereby transfer the labels of the RNA-seq to the ATAC-seq. So how do we do that? By several approaches, but they're all based largely on defining or aggregating ATAC signals with some function on the genes, so that we have a gene value for the ATAC-seq data that can be integrated with the RNA-seq, which is also a gene-based value. And then we can use NNLS or integration using Seurat for label transfer. And this way, we were able to annotate all these clusters, like the Kenyan cells here in yellow, the glial subtypes here in blue. The green are all the optic lobe clusters, and in red-brown is the central brain, which is much less annotated, as you can see. So then each cell type now, again, has a specific set of differentially accessible regions, as well as regions shared with other cell types. Here are many examples of genes and here are in rows are all the cell types where regions can be found of marker genes for that cell type that are specifically accessible there. So we could validate this as well, similar as in the iDisk, where we used driver lines, in this case for the mushroom body, and FAC sort these cells out, perform bulk ATAC-seq, and confirm that the peaks that are specifically found, for example, here in the gamma Kenyan cells, in that specific cluster, which is this cluster here, that this peak, which is specifically found there by single-cell ATAC, is also found specifically in the bulk. And this is true for these 3 cell types that we validated this way. So we are quite confident that, at least for these large clusters, our annotation is indeed correct. So what does it all mean now? So I'm going to go through a couple of examples to illustrate the data. So here's a gene called CRUMS. It's expressed specifically, or relatively specifically, in the alpha beta Kenyan cells. This is clustered in the RNA. And that particular region here, you can see the small yellow peak, I hope, is only accessible here. There's some noise here in the photoreceptors, but in the brain, it's only accessible in the alpha beta kinase cells. And that you can see here on this t-SNE of accessibility, where this particular region is shown to be accessible only there in the alpha beta kinase cells, or largely there. These are the photoreceptors here, which are contamination from the brain dissection. And so this means that we can link now this region to this gene, specifically accessible and specifically expressed. And that region could potentially regulate this gene, right? So there are, most of the cases are more complicated, as you can see here, with many to many links. This is a region that is specifically accessible in astrocytes, that you can see here, and the nearby gene. This is an intron of a gene WUN2. That is a marker of astrocytes also in the RNA. So there seem to be a lot of, well, thousands and thousands of such matches to be found. And like in the Ilis, we also tested a number of regions for enhancer activity by cloning them, making transgenic flies and checking where the GFP is expressed. And even though the success rate is a bit lower than in the Ilis we were more than 85%. Here, it's more like 60% success rate. Still, many of these peaks can be found to operate as enhancers. And there's an example here where the yellow region comes from fly light. It's specifically active in Kenyan cells. The attack peak is specifically in Kenyan cells, but it actually is much smaller than this yellow region. So what we did was to clone only the attack peak, only this 400, 500 base pair region. And then indeed, this is the region that contains the signal, the enhancer signal, to drive GFP in that cell type. So the Flylight database usually has 2-3 KB regions, but using the attack peak, we can narrow it down to where the information is actually encoded. Now, what's also interesting is that if you look in development, because remember, we had done also the developmental single cell attack. In fact, during development, this particular region is accessible in more cell types. And also, if we look at the GFP of this line, it's also active in more lines. So it's getting narrowed down to one specific cell type in the adult. Like I said, we tested many of these regions and found quite a lot of positive enhancers. Some maybe an interesting case to further illustrate is that when the region is larger, in this case, this green region here has been cloned by Janelia Farm, and it is relatively noisy. There is activity in Kenya in the optic lobe. This is an optic lobe neuron driver, optic lobe neurons, but also in glial cells. And in fact, we see that there is an attack peak in the T4 optic lobe neurons and also an attack peak in the glial cells. So in fact, if we clone only this region here and drive with GFP, then we find that it's very specific to the optic lobe neurons. So we're able to remove this signal from the glial cells. This shows that in the fly genome, it's very compact and there's a high density of enhancers. So cloning too large regions may just cover too many enhancers. Okay, so far so good. There's much more complexity, but regulatory regions are accessible in specific cell types and can function as enhancers. So we can try to build again these regulatory networks. And in this case, we are actually moving to a new version of scenic version 2, which will take RNA and attack, single cell RNA and single cell attack as input to derive these enhancer GRNs that use the co-expression of the TF with the target gene, but also the co-accessibility of the regions in that cell type, the motif enrichment of that factor in those regions, and the link between the region and the target gene. A bit similar, but a slightly more advanced as what we did in the iDisk. So here's an example of a transcription factor, Ilis. This is the PAC6 homolog that is expressed on the x-axis is expression in 4 cell types in the brain. And in the y-axis is the motif enrichment of that Ilis motif in the co-accessible regions of each cell type. So there's an agreement between these 4 cell types in terms of expression and the fact that the motif is enriched in the regions that are accessible in these cell types, while all the other cell types have neither the expression of the TF nor the motif enriched in co-accessible regions. And this way we can highlight or identify transcription factors that are important for each cell type, as shown here in this heat map. So in the Kenyan cells, we find Ilis, twin of Ilis, MEF2. And this leads us to the prediction of the key regulators, as well as where is likely binding. And then by linking those to the genes, which genes may be regulated. And then when we summarize this information, this yields the enhancer gene regulatory network, where the transcription factors are here. They are linked to these blue regions, blue nodes. And these blue nodes are the regulatory regions. So this region here is this region. So it's in the genome, it's only accessible in Kenyan cells, this left part, and Ilis is predicted to bind there. And this region is linked to this gene that is now on the outside. So the factors are inside, and the target genes are on the outside, and they are linked by the genomic regions candidate enhancers. That we have been able to derive for 40 cell types in the brain. And this is a 5-minute call? I assume. I hear a bell.
Yeah, 5 minutes left.
5 minutes left. Okay, thank you. And then this is the result of 40 different of these GRNs that we are now making available through quite an interesting website that allows, it's a bit like Cytoscape, but then in a browser, where you can actually click on nodes and regions. So clicking on this region gives information about where is that region, the genome. Can I go to the UCSC genome browser? Can check the region in scope for where it's accessible, and a link to the deep explainer neural network that I will show in a second. Indeed now. So then the final step here in the brain that we have been working on is the deep learning. So we narrowed down a little bit, because it's really extremely complex, to a subset of cell types: glial cells, T neurons in the optic lobe, and Kenyan cells in the mushroom body. And then trained again this artificial neural network using the topics from SysTopic. And on this bit limited and nice cell types, it works quite well. So we can then ask which filters were used for each cell type. So for the Kenyan cells, we find filters that are important to predict from the sequence of the enhancer where it's accessible. Filters for transcription factors, 1-cut, Ilis, MEF2, that are indeed expressed in Kenyan cells. So this is the expression of these factors. And what's interesting is that MEF2, for example, is expressed lower in the alpha prime beta prime, and the importance of the motif is also lower in the alpha prime beta prime. And then the deep learning model also delivers motifs that are negatively important. So MAMO and DATI, when this motif is present, the region will be closed in that cell type. And this MAMO is expressed in gamma and alpha prime beta prime, and that's exactly where the motif is negative. So when this is expressed, the regions with the motif will be closed. When the factor is absent, these regions will be accessible. This is another example for the T neurons, where again we find the 4-cat motif, for example, in these subtypes of the T neurons, and that is where it's expressed. So we can use the expression a bit as validation of whether these motifs make sense. All right, I have to, I'm going to skip this. These are examples of enhancers that we tested in the brain together with the deep learning explanation. And just to finish, I wanted to present a few slides on the fly cell atlas. I think it can be interesting maybe to some of you. So in the fly cell atlas, we are with a large consortium sequencing all the cells of the entire fly. And we are using nuclei extracted from all the different tissues, as well as from the entire head and the entire body. And so far, we generated, this is the first version, which is now being finalized, generated 600,000 cells and cover a large part, if not all, of the cell types in the adult fly. This is the number of cells per tissue. This is the number of genes per cell type. And here you can see them clustered by these broad categories of muscle cells, neurons, sensory organ cells, fat cells, epithelial cells, and so on. So the way that we analyzed this was using a pipeline that is available in GitHub called VIB single cell NF, or Vision, that allows us to automatically analyze each tissue in a reproducible fashion. It runs ScanPy as well as Scenic, and generates Loom files that can be opened in our viewer called Scope. And in this viewer, we used annotation jamborees with experts in the field to annotate each cell type. And this is now the result of those jamborees, where for each tissue, experts went into scope and annotated each cell type with an ontology, with the anatomy ontology. So these have IDs that can be linked in a tree of the anatomy. As you can see, a large part of the tissues could be annotated, and some parts that are gray are unknown and could not yet be annotated. So this is an example in scope. After the annotation, now you can enter a cell type like enteroblast in the gut, and then this will find you where the enteroblasts are, together with the marker genes of the enteroblasts and the annotation information. So this, we are working on a paper and on the resource that will be available hopefully this year for the community. And with that, I just wanted to conclude. I hope, I think it was fast. I tried to put quite some information here, but in half an hour, it's not so easy to explain all the methods. I hope you still get some ideas about scenic and cystopic. I explained to you how we combined RNA and ATAC-seq at single cell level in the ion tunnel disk, how we could find motifs of transcription factors in the differentially accessible regions of each cell type, and that many of them are enhancers. And the same we did on the brain. And then I showed you very briefly a vignette on the fly cell atlas. So with that, I'm thanking everybody in the lab. This is our Christmas picture that we contest that we had to make in the department. And then also our collaborators. Well, here are the key guys from the fly cell atlas. So thanks again for the invitation, and I'm open to questions.
Thanks so much, Stein. So we have a couple of questions. So the first question asks if you've tried PacBio hi-fi sequencing for single-cell RNA-seq, and if the answer is yes, how does it perform?
PacBio?
Yeah.
Long-read sequencing?
Yeah, no. That's the question.
No, that we haven't tried. There's a couple of discussions that I overheard in the lab of people who are interested in that, because we are using PacBio and Oxford Nanopore sequencing for some other projects, but we haven't tried this yet, no.
I had a question, actually. I couldn't help noticing kind of in your T-SNEs, you managed to get far better separation between your clusters using the ATAC-seq data than using the single-cell RNA-seq.
Oh, yeah?
And it did look like the cell types really clustered differently in the slides about halfway through your presentation. And so I was thinking, how much does kind of this data influence how you identify cell types in the fly-based atlas? Because obviously the big atlases, they mainly use single cell data to differentiate cell types.
Yeah, this is not so easy. So overall, I have the impression that single cell RNA-seq is still the best to find cell types. So here, it may seem that they separate a bit more, but I think it's an illusion on the T-SNE, because we find actually higher resolution of cell types, particularly in the central brain, where here we find just this continuum here, while we find many different individual clusters with the RNA-seq, which we cannot find in the ATAC-seq. There's even a cell type that we know very well, the projection neurons. I don't remember where they are here in the RNA-seq, which we cannot find in the ATAC-seq. So the resolution of finding cell types is higher in our experience with RNA-seq than with ATAC-seq. It may be due to the biology, that only a few regions may differ between some cell types that is difficult to find with this sparse method, or it may be due to the technical aspects of the single cell ATAC sparsity. And so I think ideally, and this is also where many people are moving, although it's very costly, would be to have the multi-ohm data for the atlases, where ATAC and RNA is combined from the same cell, so that if the RNA finds a cluster, then we know what the ATAC is. And then if we find a second cluster in RNA, but the ATAC is similar, we can at least do a supervised analysis of the ATAC in these two cell types, even though we wouldn't have been able, I don't know, to find these two cell types with the ATAC-seq alone. So with the combination, I think it's going to bring us really far. But yeah, we're going to have to redo everything again. if we want to have the multi-ohm there, which is it's insane, right? So these technologies are evolving so rapidly that we can almost not keep up with them.
That's good.
Yeah.
Looks like work for the human cell atlas isn't quite obsolete then.
No, it's not obsolete, but you can already see how seriously complex this gets for a simple model organism like the fly. That it is really extremely daunting, this challenge for the human, finding such information for every single cell type in the human body is, especially the brain, is going to be really, really challenging. But yeah, the human cell is also a much larger consortium with more funding from CZI, for example, than the fly cell atlas, right? So hopefully the scale will help there.